G'day, Dr. Carl Shirtloads of Science University of Sydney with part 2 of what we're supposed to be talking about with, now, what's the correct pronunciation? Professor
Carolyn Hogg.
Hogg. And we are going to get onto the biodiversity thing. We got diverted last time. So just before we dive in, you've worked in fields such as animal behaviour. You work in so many different fields, and so you're a really good role model where you can start here and follow what your natural strength is, and then reproductive biology and satellite tracking. So surely that's just lying in bed and then switching your phone and say, bring down the data from the satellite for me? No?
No, that would have been good in those days, but in the early 2000s we used to have to use good old dial-up internet.
Dial-up?
Dial-up internet, yes, in the 90s, and dial up into Telnet and download strings of information and then interpret the number strings.
Oh my God, do you have to do undo the
Take
plug,
the phone out and
phone
put the computer in the wall?
Sometimes use a screwdriver and alligator clips,
Yep.
and then watch it go, beep, 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 beep. And
That's the one.
wow.
You'll have to play the noise.
What were you satellite tracking? What creature?
So we used to use it when I was doing whale rescues to follow the whales over time. If they were entangled in gear, we would attach the satellite tracker to the entangled gear so we could follow the whales. And then later on, I worked with colleagues who were attaching satellite trackers to leopard seals in Antarctica. So I just did the data analysis for them and they did the field work.
So you could download data from the satellite. The device on the back of the animal would broadcast up to the satellite.
Yeah, and then we download
you
from
could
the
download.
satellite.
You say, okay, so you say the creature is 400 metres or kilometres in that direction.
Yeah, and then we used it when, so more recently in my career, I've been working with Tasmanian devils, and when we did some of the translocations for Tasmanian devils, we put collars, satellite collars on them so we could track them.
Now we have to
during
get
the
to
release.
that because it's part of the biodiversity thing, and also species management is another one of your fields.
Yeah, so that's around about how you manage species in breed for release programs. So how do you bring them in, breed them up, and then put them back into the wild? So the one that I've worked on, the largest program that I've worked on that most people know about is the Tasmanian devil.
Okay, so we'll just do a brief intro to make sure we get into the biodiversity
Yes,
thing, which is what we're supposed to talk about, which I did some reading on this. And apparently the idea started off in 1968 with the term biological diversity, a conference run by Raymond F. Dassman, a different kind of country, 68. 85, they had the National Forum on Biological Diversity, and the word biodiversity appeared in 1985, and then E.O. Wilson, the ad person, wrote it up in print and used the word biodiversity as a title, which, according to my little research, is the living fabric of our planet.
Yes.
And nature gives us so much public good for free. By so much, I mean trillions of dollars. We're talking several percent of the world GDP.
Yeah, so about 55% of the world's GDP.
Yep.
So that's about, I think it's 55 trillion US dollars.
Yeah, we're around 100 trillion.
Yeah,
Yep.
is moderately or highly dependent on nature. But I would actually argue that 100% of global domestic product is dependent on nature, because we depend on biodiversity and nature for the food that we eat, the medicines we get, our clean air and our clean water.
And the honey and the bees don't get paid.
And everything.
So the layers I looked at were on scales. There's local, community, national, international, and different layers such as species, ecosystems and genetics.
Yes. So biodiversity is made up of three things. So
okay.
diversity of ecosystems. So people know lots of different types of ecosystems on the planet is a good thing.
Yeah.
Anyone who did Year 5 science knows that monoculture is bad. So lots of diversity of species is really great.
Funny how they... Okay,
Yep.
keep going.
And we've
keep
known
going.
that
Yep.
diversity of genetic diversity is critically important. So it's, if you imagine a
Yeah.
tripod, you need those three things to be balanced in
And the
order
three
for the
things are?
system. Ecosystems,
Yep.
species and genes.
Yep.
And for 50 or 60 years, ecologists have done an amazing job of monitoring ecosystems and species. And we could do genetic diversity, but it was really quite hard. And then the Human Genome Project in 2003, when it was published, the Human Genome changed everything. So the Human Genome took 13 years and about, I think it's about $4 billion US dollars in today's money to do the Human
But it
Genome.
paid off so big.
Paid off so big. You think about how human medicine has changed significantly in the last 20 years. Now imagine if we created a genome for every living species on the planet. So there's a project I'm involved in called the Earth Biogenome Project, which is plans to generate genomes for all eukaryotic life on Earth. And we can do it in 10 years, same time of time for one genome for the humans. We could do all life on Earth for the same amount of money.
Hang on, say that again.
So the human genome took
Yep.
13 years and $4 billion.
Yep, and 4 billion out of 100 trillion is peanuts.
We can now do all life on Earth in 10 years
All life on Earth
for $4 billion.
in 10 years.
Yeah.
That would be such a good
And
data bank.
and you think about what we were able to do with the human genome and how it changed medicine and how we look at disease and how we investigate cancer. And then you think about having that resource for all life on Earth. So
Are we heading down that pathway?
we're trying, but it's
It's
hard to get funding for that kind of basic science.
I'd never heard of this project.
Yes. So the Threatened Species Initiative is the project that I run here in Australia.
Okay. Threatened Species Initiative.
And that project's about not only generating genomes, reference genomes for Australia's threatened species, but also just generating genetic data for different populations of threatened species. And the key point of that as well is not only about generating the scientific data, but then working with the conservation managers and the people on the ground who have to make decisions. So these are the people who, make decisions around translocations or restoration projects, the likes of the Australian Wildlife Conservancy.
Translocation.
Yeah.
Shifting an animal from one place
to another. There's lots of different translocation projects in Australia. So the Threatened Species Initiative helps generate the scientific data, and then we work with the conservation partners to improve genetic literacy to help them understand how they can use that information to inform their decisions.
Now, I'll just give two little examples to point out why we shouldn't let these species become threatened and why we shouldn't let them die out. And example number one from my notes here is the 1846 Irish potato blight, which killed 1 million people out of 8.4 million people, because while there were 13,000 species of potatoes in existence and 8,000 in common use, only not 2,000, but two species were grown in Ireland, and they fell over, and there are various other social things, and the population still hasn't gone back to 8.4 million. Example number two of the importance of biodiversity. Apparently in 1970s there was a thing called the rice grassy stunt virus, and it was just killing rice. And they tested 6,273 varieties of rice for resistance, and the number of resistant varieties they found in the whole world was one. And it was in India and known to science since 1966. So if you have those sorts of bad things potentially happening, you need the biodiversity, which does not generate profit in the next financial quarter.
Yeah.
But it does for the whole human race over a longer period of time. So tell us about this is that the right name, Threatened Species Initiative?
Yeah, so the Threatened Species Initiative I was working with the Tasmanian Devil program.
Yeah.
And we worked out, I was working in management, and Kathy, who I work with here I was at Sydney Uni.
Professor Kathy Belov.
Professor Kathy Belov, she is one of the world's leading immunogeneticists, so she discovered all the immune genes
Immunogeneticist.
for marsupials.
That's where all the clever people are in immunology.
She's very clever.
Immunology is really hard.
And so we started working together in partnership between Sydney University, the Zoo and Aquarium Association, and the Save the Tasmanian Devil program. And
that's a bunch of things you're trying
it
to
is.
bring
So
together.
we had government, we had the zoo industry, and academia.
I'm guessing you're fairly good at filling out paperwork.
Yeah,
and
and what
talking to people.
Kathy and I worked out is, if we were collecting the samples on the ground and her students and her team were producing this genetic data to do all these bigger investigations, I could use the same genetic information to make management decisions.
What? So you had this genetic information and then you could use it again.
So Kathy was
Wow.
producing it for cutting edge research in immunology and the devils and the cancer that they have.
Right.
But the same data set we could reanalyze and use it for management decisions.
For management.
And that's where we
Wow.
worked out that actually if you work in partnership between academia and industry and the conservation sector, you can accelerate the information. So I was not waiting for Kathy's students to finish and publish their papers and write their theses up, we would meet regularly and they would tell me the information before publication and I would go and use it for management decisions. And that's actually the secret to getting the translation out of academia into real world application, because the time for publication and for students is such a long period of time these days for information to go from when you do the experiments to when you actually publish the research. And it was very linear fashion. So then, you publish a paper, someone in conservation might read it if it's not behind a paywall, and then they might implement it. So start to finish, you're looking at 10 to 15 years from when the discovery was made to when the application was happening. And Kathy and I were really interested in how could we short-circuit that system? How could we make research happen in real time?
Wow.
So we started it with devils, and then we moved on to doing it with bilbies and orange-bellied parrots. And then my question to the government, so one Sunday in 2018, I read 200 vertebrate national recovery plans.
200?
200, all 200.
Hang on. If you got 8 hours, that's...
Yeah. I just skim read. I was looking for the word genetic and translocations. And of the 200 recovery plans, more than 80% of them needed genetic information for the recovery of the species.
They needed that information.
But less than 10% of them were using genetic data. And I asked government agencies, whether they were state or federal, why were they not using genetics? And it was because it was expensive and considered a nice to have, because there's a limited pool of funding available. Do you know that we only spend about 0.6 cents of every dollar of the federal budget is spent on the environment in this country? 0.6
Oh, well, luckily the
cents.
environment's no use to us and doesn't...
Well, I mean, we have a productivity roundtable and the environment is not at the table.
Keep going. Wow.
Explain that one to me.
Take it away.
A large proportion of our GDP is dependent, and our productivity as a nation and our economy is dependent on nature, but nature is never given a seat at the table. And so, what I said to the government agencies, well, if I build it, will you use it? And they said yes. And so I sought funding. We got funding support from Bioplatforms Australia, who gave us $1 million for sequencing. And we set up a collaborative program. So the Threatened Species Initiative is not just Sydney Uni. It is a collaborative initiative across the country. There's about 270 collaborators now involved in it. And in 4 years, we thought we would do 40 species. And in 4 years, we've done 125 species.
Wow.
So there's 125 threatened species in Australia today that now have genetic information that can be used for management decisions, whether it's translocation, restoration, creating seed banks or biobanks or captive breeding programs that never had data 4 years ago.
Wow. That sounds like a huge body of work you've done.
I know, and I've just got some more money, so now I'm going to go for more species.
Okay, so 125 species-ish, you got the genome. What can you do with it once you've got that info?
Yeah, so that's a commonly asked question. So in our research group, we specialise in wildlife with disease. So we work on Tasmanian devils, we work on koalas with chlamydia, we work on frogs and chytrid. And so what we're trying to understand is how much genetic variation does a species need to have to be able to start to evolve and adapt to a disease. And wildlife diseases are becoming more prominent, not just because we're getting better at detecting them, but also as you reduce habitat, you're pushing together species that never co-evolved. So that's where new diseases are coming from. So that's where COVID came from. You take species that have evolved in different types of environment. They no longer have any habitat to live in. They're now all crammed in together in these tiny little pockets of habitat that we have across the planet. Because in my personal opinion, there's no wild left. The wild is gone. It is just pockets of habitat fragmented by humanity.
Because wild involves communication.
And you need movement and you need connectivity in the system. And so, species' immune systems co-evolve with the pathogens that they're exposed to. So when something new comes along, they don't have capacity to adapt. And so a genome is like having the puzzle box lid. If you were going to do a 3.5 billion piece jigsaw puzzle, that kind of looks like all the jelly bean jigsaw puzzle. Let's make it a little bit harder for you. We'll give you colours. We won't make it all the same colour.
Okay.
Is it easier or harder to do if you have the puzzle box lid?
I need the puzzle box lid, please, can I?
Yeah. So a reference genome is a puzzle box lid. So when we sequence genetics, when you hear people say, oh, we did genetic sequencing, we're not sequencing all human genomes, 3.2 billion pieces. So if you imagine DNA is a ladder, and each rung of the ladder, there's 3.2 billion rungs in the human genome. There's 3.7 billion rungs in the bilby genome.
Really?
The corroboree frog, you know, the tiny itty-bitty little corroboree frog, the black and yellow one that's found in Kosciuszko.
No, I know.
10 billion rungs in the...
What is in there?
Yeah.
Wow.
Lots of repeat
Because
regions.
I remember at one stage we had something called the gastric brooding frog, which could turn its stomach, which is normally full of acid, into a uterus.
Yes.
Which meant it had to switch off this acid production, and we're talking in human stomach ulcers, and yet we wiped it out.
Yes, we did.
That secret's gone forever.
Yeah.
Oh my God.
And so that's what we use the reference genomes for. So when we sequence, we just have all these different pieces of the puzzle, but because we've got the lid, we know exactly which piece of the puzzle we're looking at and what it does. So we specialise in immune genes. There's other research groups that look into reproductive genes. We also use genetics to tell populations apart, to tell species apart. We can look at historical gene flow. So we just did the koala genome survey. So we sequenced 470 genomes. So we sequenced the whole genome, 3.2 billion pieces of 470 koalas, all the way from far north Queensland right through into South Australia. So for the first time now, we have a map, a genomic map of koala genetics across the range, and we can look at differences in their reproductive genes, in their immune genes, what gives them fluffy ears. That's what Kathy really wants us to work on.
Fluffy ears?
Yeah, why do koalas have fluffy ears in the south more than the north? And these are important questions because, you know, with climate change coming and the range of koalas is getting restricted because the western part of their range is drying out. Mm-hmm. So there's not enough water for the trees.
Really, the western part of their range is drying out.
Koalas are getting heat stress. Koalas really love to live where humans live, but because we've got such significant development along the east coast of Australia now, we've taken out huge portions of koala habitat. But we can look at the genomes. It tells us what happened in time. So we can go back about 20,000, 30,000 years with demographic modelling, looking at what happened with koalas 20,000 years ago by sequencing a genome today. So genomes are just they're a massively powerful tool. And, you know, we know from the Human Genome Project what you can do for human medicine with a genome. But with wildlife genomes, it's just this untapped resource that once you've generated it, you can just keep going back and mining the data set for quite a long period of time.
So you've got this group of 120-something species in Australia, and they're all threatened, are they?
They're all threatened, yeah. Some are extinct in the wild. We have two extinct in the wild species in Australia. Did you know that?
No.
The Christmas Island blue-tailed skink and the Lister's gecko, both from Christmas Island, were made extinct by the wolf snake, and they were brought into captive breeding programs. I think there was about 67 from memory skinks and about 40 Lister's geckos. We work with Taronga Zoo, who breeds them, and Parks Australia on Christmas Island, and we've done all the genetics for them to try and help their breeding program to maintain the diversity of those species. Because once they go extinct, they're gone forever.
So with that exact word diversity, how much in that 120-ish number of species, how many are widely diverse in their genome, and presumably that gives them better survival, and how many of them are narrowly diverse? I kind of think that, is the cheetah fairly narrowly diverse? They went through a breeding loophole.
Cheetahs have low diversity. So in Australia, of our Australian species, Tasmanian devils actually have the lowest genetic diversity I've seen out of anything. And the reason is because they've gone through about 3 or 4 population crashes and severe bottlenecks over 20,000 years. And we see this
over 20,000 years.
And they've had this long, slow decline. And so when you have the long, slow decline, every generation is losing diversity, and so that overall individuals have lost diversity. In the species where you've had large populations that drop off really rapidly, the few individuals you have left can still have lots of overall genome-wide diversity. So
after the first population crash.
So after the first if it's sharp and quick, the danger is populations staying small and breeding at very small levels for a long period of time, because then your only option is to breed with relatives, and that's where you lose genetic diversity. So the secret to threatened species is if they fall off the cliff is to provide the opportunity for them to breed rapidly, so population grows quickly, so you can maintain genetic diversity. Because otherwise you can end up with a large population with low genetic diversity, which is what happened with Tasmanian devils. And then something comes along, such as the infectious cancer they have, and they can't mount an immune response to it. And so now it's taken out 80% of the species is now gone.
80%?
Of devils have been lost in the last 20 years because of devil facial tumour disease.
And they've got this transmissible
They've got
cancer?
two.
Two called... Oh, two. I didn't know that. So one of them's called...
They're both called, and we're very original, devil facial tumour disease 1 and devil facial tumour disease 2.
Okay. So you're doing what sort of seems like good work. But why should we do this? What's the motivation? What's the eventual outcome of this?
I guess for me there's two eventual outcomes. I'd like to think that when I'm old and just about to kick the bucket, I can look at my grandchildren squarely in the face and my great-grandchildren and say, I actually tried.
I done good.
To try and save some species. You know, you guys get to see these species on the planet because of the work that I did. But on a more philosophical nature, we are not separate from nature. There's 12 million species on the planet. And
12 million?
We think, roughly. We're not even sure how many species
Of
are in Australia.
life or above in the air and the water, or is that total altogether? Or?
That's all together, we think.
We think ish.
We think ish.
Okay.
And humans are one species on the planet. We're not separate. It's not like there's nature and then there's humanity. It's all in one. So, you know, as I said earlier, our food comes from the environment. The medicines that we take come from the environment. Our clean air, our clean water, our clean air is all because, you know, you've got trees cleaning the atmosphere, and the water systems are coming from clean waterways. If you don't have those systems, where do you think humanity is going to end? And so although, you know, for decades, all the way through like the 20th century and into the 21st century, we've had this mentality that the earth is limitless, but we now know it's not. And so we need to get better at thinking around, you know, when I say to people who work in business, who likes to go out in nature, and everyone will put their hand up. And then it's like, who knows nature is important for their business? And most people recognise that their business has some touch point with nature. Who thinks nature is a responsibility of government? Everyone leaves their hands up. Who wants to pay more tax? And everyone puts their hand down. So we know we want it, we like it, we need it, our lives depend on it, but we do not want to pay for it. And unfortunately, we are now in a situation where unless we start thinking about how we can truly invest in nature and not just 0.6 cents of every dollar, but actually make proper investment, the systems are going to keep crashing, and food will become more expensive, and clean water will become harder to get, and clean air will become a thing of the past.
Wow.
So where do people want the story to end?
With a happy ending?
Hopefully with a happy ending. I like to think that there's people like me who are fighting the good fight, but it's no longer the realm of just people that work in conservation or people that just work in biodiversity. I think there needs to be a wholesale change. And society is at that point of change, but for some reason there seems to be a disconnect between what society wants and what the policymakers will do.
Is that the balance between short-term private profit and long-term public good?
I don't even think it's private profit. I think it's just any kind of profit.
Short-term profit
and long-term good. And it's not, you can, this is a space I work in, is like, how do we still have agriculture and farms to feed the world, but farm in such a way that allows us to protect biodiversity? How do we build cities that don't cause us to raise the entire property to the ground and take out every tree for miles? How do you build built environments where you can maintain aged old trees? We need to just get smarter at the way we do things rather than just bulldozing it all to the ground in order to put up, some houses and then plant some trees which take 40 years to grow.
On the positive side, how can people follow you in your fine work and become your PhD students or attend your lectures here at the University of Sydney, where you are a professor?
So I'm on LinkedIn. That's where I put
LinkedIn.
most of my stuff these days. We also have a website, which is the Wildlife Genomics Group at Sydney Uni. You can just Google that. That brings us up. We have we get our students actually write a lab blog, so they tell you about their projects and what it means to work on the multitude of different species that they work on.
Thank you so much for blowing my mind and telling me about your amazing career.
Oh, thank you so much for having me. It's been joy.
Thank you, Professor Carolyn.
Great, thank you.
Shirtloads of Science is washed, spun and aired by the University of Sydney.